The two SoftChalk lessons for Unit 2, Lecture 3 deal with ways in which the chemical control agents we use affect bacteria. Now, before we get into the way antibiotics and such work against bacteria, let's have a little overview of the control of microorganisms with some definitions we'll need to know from here to the end of Unit 2. First of all, some bullet points or fundamental statements. Physical control includes methods of control as high or low temperature, desiccation, osmotic pressure, radiation, and filtration. Now, for my class, we're going to cover physical control in lab, as you'll notice in your table of contents for your SoftChalk lessons. So you won't be responsible for physical control on the lecture exam, but rather later on a lab quiz. Chemical control refers to the use of disinfectants, antiseptics, antibiotics, and chemotherapeutic antimicrobial chemicals, which we will be covering here. Some definitions. Sterilization is the process of destroying all living organisms and viruses. Disinfection is the elimination of microorganisms, but not necessarily endospores, from inanimate objects or surfaces. Decontamination is the treatment of an object or inanimate surface to make it safe to handle. A disinfectant is an agent used to disinfect inanimate objects, but is generally too toxic to use on human tissue. An antiseptic is an agent that kills or inhibits the growth of microbes, but is safe to use on human tissue. A sanitizer is an agent that reduces microbial numbers to a safe level. An antibiotic is a metabolic product produced by one microorganism that inhibits or kills other microorganisms. Synthetic chemicals can be used therapeutic as well. They're called synthetic chemicals. An agent that is cytal in action kills microorganisms, whereas an agent which is static in action inhibits the growth of microorganisms, inhibits them from growing or slows them down. Selective toxicity means that the chemical being used should inhibit or kill the intended pathogen without seriously harming us, the host. A broad spectrum agent is one that's generally effective against a variety of gram-positive and gram-negative bacteria. A narrow spectrum agent generally works against just gram-positives, just gram-negatives, or only a few bacteria. So those are bullet points on some definitions and your detailed learning objectives for this short SoftChalk lesson. Define the following: selective toxicity, broad spectrum antibiotic, narrow spectrum antibiotic, chemotherapeutic synthetic drug, cytal, static, sterilization, disinfection, disinfectant, antiseptic, and physical agent. So, of course, control of microorganisms is often essential in preventing the transmission of disease and infection. We use it to stop decomposition and spoilage, to prevent unwanted microbial contamination. So, microorganisms can be controlled by both physical agents and chemical agents. Physical agents include such things as high and low temperature, desiccation, osmotic pressure, radiation, and filtration, anything not chemical. Chemical agents refer to the use of disinfectants, antiseptics, antibiotics, and chemotherapeutic antimicrobial chemicals. Although you need to know what a physical agent is by definition, physical agents we'll be covering later on in lab rather than in lecture. And here's some terminology we'll need to know for control using chemical agents. Now, sterilization is the process of destroying all living organisms and viruses. So a sterile object is one that's free of all life forms. That includes endospores as well as viruses. Disinfection is the elimination of microorganisms, but not necessarily endospores, from inanimate objects or surfaces. Decontamination is the treatment of an object or inanimate surface to make it safe to handle. A disinfectant is an agent used to disinfect inanimate objects, but is generally too toxic to use on human tissue. Whereas an antiseptic is an agent that kills or inhibits the growth of microbes, but is safe to use on human tissue. A sanitizer is an agent that reduces microbial numbers to a safe level. An antibiotic is a metabolic product produced by one microorganism that inhibits or kills other microorganisms. So we do initially get our antibiotics from other living organisms, although they're often extensively modified and some are synthetically made today. Chemotherapeutic synthetic drugs, these are synthetic chemicals that we use to treat therapeutically rather than made by microorganisms. An agent that is cidal in action will kill microorganisms and viruses, whereas an agent which is static in action will inhibit the growth of microorganisms, slow down their growth, but not necessarily kill. So anyway, those are some general definitions. A number of other definitions we need when we talk about antibiotics shortly are listed here. Now, the whole basis of chemotherapeutic control of bacteria, that is using chemicals to treat an infection, is selective toxicity. So selective toxicity means the chemical we're using should inhibit or kill the intended pathogen without seriously harming us, the host. So keep in mind, although antibiotics are some of the safest drugs we have, people do die from taking antibiotics. People do have serious mishaps from taking antibiotics. No drugs that we use are 100% safe. A broad spectrum agent, like a broad spectrum antibiotic, is generally one effective against a variety of gram-positive and gram-negative bacteria. Works against a wide range or broad spectrum of microbes. Whereas a narrow spectrum agent, like a narrow spectrum antibiotic, works against just gram-positives or just gram-negatives, or maybe only a few bacteria. Most of our anti-tuberculosis drugs are very narrow spectrum. They generally only work against Mycobacterium tuberculosis, for example. And again, these chemical agents we use to treat infection may be cidal or static in action depending on the agent we're using. Remember that a cidal agent kills the organisms, whereas a static agent inhibits growth long enough for the body's defenses to catch up and remove the organisms. So for example, drugs that inhibit peptidoglycan synthesis causing osmotic lysis are cidal in action because the bacteria burst, they're killed. But a lot of the antibiotics that bind to ribosomes bind reversibly to ribosomes. And so if they bind reversibly, that means the drug attaches and it comes off and it attaches and it comes off. And so they don't kill the organism, but they inhibit it. They slow it down. And often that's all we need to do for the body's defenses to be able to remove what's left. And there are two categories of our antimicrobial chemotherapeutic agents: antibiotics and synthetic drugs. Now, antibiotics are traditionally defined as metabolic products of one microorganism that inhibit or kill other microorganisms. So we do know there's some microorganisms out there in nature that secrete these chemicals that inhibit or kill surrounding microbes to get rid of their competition. And we discovered that by accident, but that gave us one of the first ways to actually be able to treat infectious disease. And then chemotherapeutic synthetic drugs are antimicrobial drugs synthesized by chemical processes in the lab. But the distinction between these two aren't all that distinct today because many of today's antibiotics are semi-synthetic and some are even made synthetically in the lab. But traditionally, antibiotics were originally derived from other microorganisms. So those are the key definitions we'll need to be familiar with as we go through this next section or two. And I'm combining these two SoftChalk lessons together since that first one is so short. So we're going to start out by looking at the ways in which the chemical agents we use affect bacteria. And in the following lecture, we'll be looking at how bacteria become resistant to these chemical control agents. So our goal in this SoftChalk lesson is to look at the various ways in which our chemical control agents work. What are they doing to the bacteria to inhibit or kill them? So let's start out with our fundamental statements or bullet points. Many antibiotics like penicillins, cephalosporins, vancomycin, and bacitracin inhibit normal synthesis of peptidoglycan by bacteria, causing osmotic lysis. And they do this by inactivating the enzymes or the transporters involved in peptidoglycan synthesis. A few antimicrobial chemotherapeutic agents, such as INH and ethambutol, inhibit normal synthesis of the acid-fast cell wall. A very few antibiotics like polymyxin, colistin, and daptomycin alter the bacterial cytoplasmic membrane, causing leakage of molecules and enzymes needed for normal metabolism. Some antimicrobial chemotherapeutic agents like the fluoroquinolones, sulfonamides, and trimethoprim inhibit normal nucleic acid replication in bacteria. Many antibiotics like tetracyclines, macrolides, oxazolidinones, and streptogrammins alter bacterial ribosomes, interfering with the translation of messenger RNA into protein and causing faulty protein synthesis. There are two common antimicrobial modes of actions for disinfectants, antiseptics, and sanitizers: those that damage lipids or proteins of the cytoplasmic membrane, resulting in leakage of cellular needs, and those that denature microbial enzymes and other proteins. A number of factors that influence the antimicrobial action of disinfectants and antiseptics, including the concentration of the chemical agent, the temperature at which it's being used, the kinds of microorganisms present, the number of microorganisms present, and the nature of the material bearing the microorganism. All of those influence the action of disinfectants and antiseptics. And endospore producers such as Bacillus species and Clostridium species and acid-fast bacteria like Mycobacterium tuberculosis are harder to eliminate. Now, I should point out that the last several objectives on using disinfectants, antiseptics, and such to kill, you'll notice there's no objectives down here on those because we're going to cover that more in lab. But the section on antibiotics and antimicrobial chemotherapeutic chemicals, the first 5 bullet points, we definitely will have on the next unit exam, and that covers these detailed learning objectives. So after this SoftChalk lesson, you should be able to describe 6 different ways antibiotics or disinfectants may affect bacterial structures or macromolecules, and state how each ultimately causes harm to the cell. State which of the following groups of antibiotics inhibit peptidoglycan synthesis or inhibit nucleic acid synthesis or alter the 30S ribosomal subunit blocking translation or alter the 50S ribosomal subunit blocking translation. And here's the groups. If you have this whole list, like the macrolides, the oxazolidinones, and all of those listed as part A, be able to match those up with their mode of action. Likewise, B, penicillins, monobactams, carbapenems, etc., and match that up with the mode of action. So for example, these are all ones that would inhibit peptidoglycan synthesis. So again, if I give you the list, you match it up with the mode of action. So there's a number of modes of actions for antibiotics and chemotherapeutic synthetic chemicals that we use therapeutically. And as we pointed out back in Unit 1, many of these antibiotics inhibit normal synthesis of peptidoglycan by bacteria, causing osmotic lysis. So remember, for selective toxicity to occur, for the drug to inhibit or kill a microorganism without harming our cells, we try to find some chemical that will bind to something a bacterium has that a human cell doesn't have. And obviously those drugs that inhibit peptidoglycan synthesis are ideal because, of course, human cells don't have a cell wall, and of course we never have peptidoglycan, the component of the cell wall of bacteria. So keep in mind, as we learned in Unit 1, for bacteria to increase their size following binary fission, they have to break down the existing peptidoglycan, insert new peptidoglycan monomers so the cell can grow, and then hook it back up together again for everything to be resealed. And this is done through a series of enzymes. Remember, the autolysins are the ones that break the glycosidic bonds between the peptidoglycan monomers at the point of growth in the existing peptidoglycan, whereas other autolysins are going to break the peptide cross bridges that link the rows of sugars together. And there's a little animation that we have the autolysins that break the glycosidic bonds between the peptidoglycan and the row, and those that break the peptidoglycan crosslinks between the rows. Then bactoprenols are going to assemble peptidoglycan monomers, transport them across the cytoplasmic membrane to insert them into the existing peptidoglycan. So again, the various components of the peptidoglycan monomer, which remember is NAG, NAM with a pentapeptide, these assemble on bactoprenol. And then the bactoprenol is a transport protein that transports them across the membrane so they can be inserted into the cell wall. Then transglycosylase enzymes insert and link the new peptidoglycan monomers into the breaks in the peptidoglycan. They form the glycosidic bonds that connects one sugar in the row to the next sugar. So they're going to form these bonds here that insert the peptidoglycan monomer into the existing peptidoglycan in the row by forming glycosidic bonds, transglycosylase. And finally, transpeptidase enzymes reform the peptide crosslinks between the rows and layers of peptidoglycan. So again, transpeptidase is going to produce a short chain of amino acids or a peptide that connects one row to the next row. And that, of course, is why we learned the details about peptidoglycan synthesis back in Unit 1. And I've included the animation on peptidoglycan synthesis, so if you need to review that or you're a little fuzzy on that, make sure you look at this animation again. But anyway, the idea here is that by interfering with forming a proper cell wall, a weak cell wall is created. And remember, the peptidoglycan is what prevents osmotic lysis of bacteria under their normal growth conditions. And so there's a number of generic groups of drugs that do that, and many different versions in that group, from the penicillins, the cephalosporins, carbapenems, monobactams, and glycopeptides like vancomycin. These all inhibit peptidoglycan synthesis, causing osmotic lysis. Now let's take the example of the penicillins and the cephalosporins, two of our most widely used groups of antibiotics. Penicillins and cephalosporins, as well as other beta-lactam antibiotics, bind to the transpeptidase enzyme. In fact, that enzyme is sometimes called the penicillin binding protein because that is the enzyme that penicillin binds to inhibit peptidoglycan synthesis. But remember, transpeptidase enzyme is responsible for reforming the peptide crosslinks between the rows and layers of peptidoglycan to make the wall strong. So if the transpeptidase doesn't cross-link the rows and chains, then there's a weak cell wall, and as water flows into the bacteria, as it normally does in a hypotonic environment, the bacteria burst from osmotic lysis. So these, in fact, are cidal in action. They kill bacteria because the bacteria burst. Of course, the bacteria have to be actively dividing for these to work. If the bacteria aren't dividing, they aren't making cell wall, and the antibiotic has no effect until they start dividing again. And so we showed you an animation on that, but let's take a look at that again. So when we look at peptidoglycan synthesis, this is the new peptidoglycan monomer that's been inserted during cell wall synthesis. In this row, this is the new peptidoglycan monomer that's been inserted in this row. So once the monomers are inserted, and once transglycosylase hooks them into the row, which has already occurred here, then transpeptidase is going to connect the peptides coming off of one NAM with the peptides coming off another NAM to form the peptide cross-link that gives the wall its chain link type appearance of a chain link fence and makes the wall really strong. And during the process, one of the amino acids comes off and provides energy. So you see what transpeptidase does, as we see here. It forms that peptide cross-links between the rows and between the layers to finish sealing up the cell wall and making it strong. Now, what penicillin does and what cephalosporins do is they bind to transpeptidase. And when the drug binds to the enzyme can no longer form the cross-links between the rows and between the layers of peptidoglycan. So now the wall is relatively weak because it lacks these peptide cross-links. And as autolysins continue to break down the cell wall, but no cross-links form, eventually the wall becomes weak and the bacteria burst from osmotic lysis. And here we see the effect on bacteria exposed to a penicillin. So these are sensitive and these are resistant. And you see when there's osmotic lysis, as all the cytoplasm flows out, that just leaves the remains of the cell wall. These are resistant to penicillin, so as they divide, they don't burst. So here we see the osmotic lysis occurring. And in the second one, you can actually see the cytoplasmic membrane balloon out and burst as the cell wall becomes weak. So you see it growing. So there's the membrane coming out. So you see the membrane bulging out, and then all the cytoplasm flows out. Another group of antibiotics that inhibit peptidoglycan synthesis are the glycopeptides. Vancomycin is a nice example of that. And this actually binds to two of the amino acids on the pentapeptide chain that comes off the NAM. So rather than binding to transpeptidase, it actually binds to the pentapeptide of the new monomer. But then when transpeptidase tries to hook them together, it can't hook them together because, again, the drug is bound to the amino acids, so the amino acids can't join to the peptide chain. And again, you have a weak cell wall and they burst. So we see that in this animation. So once again, the peptidoglycan monomers have been inserted into the row by transglycosylase. The final step is that transpeptidase is going to connect the peptides coming off of one NAM with the peptide coming off the other NAM in a different row. And that's the peptide crosslinks that make the cell wall strong. So again, that's what transpeptidase normally does. Now, vancomycin doesn't bind to transpeptidase, but rather vancomycin binds to the amino acids coming off the NAM. but once they bind to the monomer, transpeptidase can't cook them together. The drug's preventing this peptide chain from being connected to this peptide chain. So again, there's no peptide cross-link formed because the vancomycin's in the way. And as autolysins continue to break down the cell wall, but no peptide cross-links form, eventually the wall becomes weak, the cytoplasmic membrane balloons out, and the bacteria burst. And then bacitracin, often found in the triple antibiotic creams you can buy over the counter, binds to the transport protein bactoprenol. And when it binds to the bactoprenol, that prevents it from assembling the peptidoglycan monomers. So in this case, there are no peptidoglycan monomers assembled onto the bactoprenol to be transported across the wall and incorporated into the growing peptidoglycan. And we see that in this animation. So remember, normally the peptidoglycan building blocks or peptidoglycan monomers assemble on bactoprenol. First 5 amino acids are added to the NAM to form the pentapeptide. Then an energy compound connects that to the first phosphate on bactoprenol. Then NAG is attached to the NAM. Now you have the complete monomer. And then bactoprenol transports that across the membrane and assembles a new monomer, and will eventually transport that across the membrane, working over and over again all around the bacteria. So that's the job of bactoprenol, to assemble the monomer and transport it across the cytoplasmic membrane where the peptidoglycan is located. Now what happens with bacitracin is that as the bactoprenol is getting ready to assemble a new monomer, bacitracin will attach to that. And once the bacitracin attaches, the NAM can attach, so there's no monomers assembled and there's no monomers to be transported across the membrane. And of course, as autolysins continue to break down the cell wall, there are no peptidoglycan monomers being transported across, the wall becomes weak, and we have osmotic lysis. So those are antibiotics that inhibit peptidoglycan synthesis, causing osmotic lysis. And this link here takes you to a list of a whole bunch of different antibiotics, just to remind you. So these are the ones that inhibit peptidoglycan synthesis, causing osmotic lysis: the penicillins, the cephalosporins, carbapenems, monobactams, glycopeptides, bacitracin, etc. And there's a little self-check on those that inhibit peptidoglycan synthesis. Now we have a few antimicrobial chemotherapeutic agents that inhibit synthesis of the acid-fast cell wall, which means mainly the genus Mycobacterium, used mostly for Mycobacterium tuberculosis, which of course causes tuberculosis or TB. So INH, or isoniazid, one of the standard drugs used in tuberculosis, appears to block the synthesis of mycolic acid, a key component of the outer membrane of the acid-fast cell wall. So remember, the outer membrane of the acid-fast cell wall is composed mostly of a waxy lipid called mycolic acid, and INH appears to interfere with the synthesis of that. And then ethambutol, another anti-tuberculosis drug, also interferes with the synthesis of some of the lipids in the outer membrane. And the anti-tuberculosis drugs in our table here are down at the very bottom. Notice all the antibiotics we have against most of the common bacteria. And then down here we have the anti-tuberculosis drugs, just a small list, because most of the antibiotics, remember, won't enter the acid-fast cell wall. So we only have a few drugs really effective against Mycobacterium tuberculosis. So these are quite narrow spectrum. There's a few antibiotics that alter the bacterial cytoplasmic membrane, causing leakage of molecules and enzymes needed for metabolism. So the bacteria don't burst, but various nutrients leak out. So the few antibiotics, things like polymyxin, colistins, and daptomycin work this way. Now, quite a few of our disinfectants and antiseptics work this way. Things like orthophenylphenol, chlorhexidine, hexachlorophene, zephyrin, alcohol, triclosans that used to be in all the antibacterial soaps before FDA stopped allowing them to put that in. These all alter the cytoplasmic membrane, causing leakage. So that includes the polymyxins, daptomycin, and pyrazinamide. Pyrazinamide interferes with fatty acid synthesis in the membrane of Mycobacterium tuberculosis. Polymyxins alter the outer membrane of gram-negative bacteria, and daptomycin also appears to bind to the membrane, interfering with normal function. So not too many there because membranes aren't all that different in nature, but we do have a few that we use against the bacterial cytoplasmic membrane. So as we see in our list here, these are the ones that alter the cytoplasmic membrane, like polymyxins and daptomycin. There are some antibiotics and synthetic drugs that inhibit nucleic acid replication in bacteria. And the primary group, which we talked about back in Unit 1 when we looked at the chromosome and the topoisomerase enzymes, are the fluoroquinolones, a whole group of drugs that have an oxygen or a floxacin in their name, such as ciprofloxacin, sold under the brand name Cipro. Now, these work by inhibiting one or more of a group of enzymes called topoisomerases. And remember, these are the enzymes needed for supercoiling and then uncoiling the DNA so it can replicate and produce a complementary copy, and so that transcription can occur. So because the bacterial DNA is circular, these topoisomerase enzymes, by putting cuts and then joining them back together again, will cause that very huge long molecule to supercoil where it'll fit. But then when the DNA gets ready to replicate, DNA polymerase has to attach, so it has to uncoil for that to attach. And during transcription, RNA polymerase has to attach to a gene, so again, it has to uncoil to do that. So these topoisomerase enzymes catalyze the supercoiling and then the uncoiling of the DNA during normal DNA replication, during DNA repair, and during transcription of DNA into RNA. So these are examples, again, of drugs that bind to an enzyme, in this case, topoisomerase. And the idea behind any of these antibiotics that bind to bacterial enzymes is that, remember, in this case, it's working because the enzyme has two specific shaped sites on it. The active site, which is where the substrate binds to the bacterial enzyme so the bacteria can carry out its normal chemical reaction. But it also has another shape on another part of the molecule called the antibiotic binding site. And that has a shape that the antibiotic happens to fit. So as long as there's no antibiotic bound to the antibiotic binding site, the enzyme's active site is fully functional, and the enzyme carries out its normal reaction and does whatever it's supposed to do. And in this case, it would carry out its topoisomerase activities. Now keep in mind the transpeptidase that we just talked about is another example. That's an enzyme, and when the drug binds to it, it alters the active site. So again, that's working the same way. But in the presence of the antibiotic, if the antibiotic's there, the antibiotic is able to fit the antibiotic binding site. And once the drug binds to the enzyme, that distorts the shape of the enzyme, alters the active site so that it can no longer carry out the normal chemical reaction it's supposed to carry out for the bacterium. And so in this case, the drugs are binding to the one of the topoisomerase enzymes. Now, another group of antibacterials that we can use to inhibit nucleic acid replication are the sulfonamides, and also another small group, diaminopyrimidines. There's only one example of that, trimethoprim. And usually these are combined together. One of the sulfonamides, usually sulfamethoxazole, is combined with trimethoprim. And these two drugs together block two of the enzymes in a bacterial pathway needed for bacteria to make a cofactor called tetrahydrofolic acid. Now, tetrahydrofolic acid is a cofactor bacteria need in order to synthesize nucleotide bases like thymine, guanine, uracil, adenine. As we see in figure 11 here, bacteria have to synthesize tetrahydrofolic acid in order to make most of the purine and pyrimidine bases that are the building blocks for DNA and RNA. And to do this, they take a chemical in their diet called para-aminobenzoic acid and use one enzyme, pteridine synthetase in this case, which you don't have to know, to convert that to glutamic acid. Then they use a second enzyme in red to produce dihydropteric acid. Then they use a third enzyme to convert that to tetrahydrofolic acid. So it takes those three enzymes for bacteria to make the tetrahydrofolic acid, which they have to make in order to synthesize thymine, guanine, uracil, and adenine. So it turns out the sulfonamides, the first of the drugs here, bind to the first enzyme in the pathway. And remember, if the drug binds to the enzyme, that alters the active site so the enzyme doesn't function. And then the trimethoprim binds to the third enzyme in the pathway. So by tying up two of the three enzymes in the pathway, that prevents the bacteria from synthesizing tetrahydrofolic acid, and without that, they can't make the building blocks for DNA and RNA. And these two are often combined under the brand name Septra or Bactrim. And they work through a process called competitive antagonism. So the idea here is the drug we're using chemically resembles a substrate in one of the bacterium's metabolic pathways. So here's a bacterial enzyme. This is the substrate that would normally bind here, but the drug has a similar shape to the normal substrate. And so either the normal bacterial substrate the bacteria wants to use or the similar looking drug can bind to the enzyme. And remember, enzymes are present in cells in small amounts. So if you have enough drug inside the bacterium, you can tie up all the enzyme, and that prevents the substrate from attaching, and that blocks the chemical reaction. But the drug's competing with the substrate for the limited enzyme. And again, that's how sulfamethoxazole and trimethoprim work. Now we can see an animation of competitive antagonism. So as we see in this animation, there's the bacterial enzyme. In this case, maybe it's one of the enzymes used to make tetrahydrofolic acid, like this first one here. So the enzyme can either bind to the normal substrate, shown in red, or to the drug, shown in blue. Now, if the normal substrate binds, like we see here, then the normal chemical reaction occurs. But if the drug binds, that prevents the substrate from binding, and that ties up the enzyme so the enzyme can't function. So the idea here behind competitive antagonism is that there's enough drug inside the bacterium that virtually all of the limited bacterial enzyme winds up being attached to the drug and unable to attach to the normal substrate. So the drug is resembling a substrate, and it competes with the substrate for the enzyme. If there's enough drug present, it ties up pretty much all of the enzyme, blocking that enzyme's normal function. And our third group of drugs that interfere with nucleic acid replication, it's called metronidazole. And this is a drug that's actually activated by a couple of proteins found only in microaerophilic and anaerobic bacteria and a few anaerobic protozoans. Remember, microaerophilic bacteria only grow if there's very small amounts of oxygen present. And of course, obligate anaerobes grow only without oxygen. And so these organisms, proteins in these organisms called flavodoxin and ferredoxin are activated by the metronidazole, and when activated, that puts nicks in the microbial DNA strands. It breaks the strands of DNA. So these are used against some anaerobic bacteria and also against a couple of protozoan that happen to carry out fermentation. They don't have an electron transport chain. And then we'll have one group of drugs, rifampin or rifamycin, and this blocks transcription by inhibiting RNA polymerase, the enzyme that transcribes DNA into messenger RNA. So if we look at our various drugs again, move down, these are the ones that inhibit nucleic acid synthesis. Most of them bind to the topoisomerase enzymes, the fluoroquinolones, for example. Some prevent synthesis of tetrahydrofolic acid, like the sulfonamides and trimethoprim. Some drugs, when activated, put nicks in the DNA, like metronidazole, and then finally those that inhibit RNA synthesis by binding RNA polymerase, rifampin. Now, another target that we favor for bacteria, of course, are bacterial ribosomes. Because remember, bacterial ribosomes are composed of 50S and 30S ribosomal subunits, whereas our ribosomes are composed of 60S and 40S subunits. And by having a different S value, again, that tells us that although the ribosomes in bacteria and in human cells are carrying out the same function, the ribosomal subunits have different shapes because they have different orders of nucleotides, because they have different genes coding for them. And so something that will bind to a 50S ribosome won't bind to a human ribosome. Something that binds to a 30S ribosomal subunit won't bind to a human ribosome. So these are very good in terms of selective toxicity. And of course, by altering the ribosome, that's going to interfere with translation of messenger RNA into proteins, resulting in faulty protein synthesis, so structural proteins and enzymes aren't made. And a lot of how these work depends on where they bind on the ribosome. There's a little animation you can use here if you want to review that on normal translation, showing you the steps in translation that we learned or reviewed actually for your molecular genetics review exam that you took home. So if you want to review that, you can, but let's look at some of the proposed mechanisms for how some of these antibiotics work. The aminoglycosides bind irreversibly to a 16S RNA in the 30S subunit of a bacterial ribosome. And there's a couple theories as to how this works. Now, we should point out that these are pretty toxic antibiotics that can cause more problems in humans than most. They're generally only used when first-line antibiotics are not effective and it's a life-threatening infection. But there's a couple proposed mechanisms that we'll share you in animations. One way is that we think some of the aminoglycosides, and as we said, they bind to the 30S subunit, the small one down here. And when the antibiotic binds, shown there as a brown oval, it's thought to kind of block the messenger RNA in the ribosome. Now remember, once the amino acids have been attached from the transfer RNA in the P site to the newest transfer RNA on the A site, that's called a peptidyl transfer RNA, then as the RNA moves down the ribosome, that causes the transfer RNA over here on the A site to move to the P site. And that opens up the A site so the next codon can be read. Well, we think the drug prevents the transfer RNA on the A site from ever moving to the P site. It basically gets knocked off the ribosome. And so that's going to prevent, again, protein synthesis. And let's skip down to that animation and show how that might work. So here's one of the proposed mechanisms. The aminoglycosides might bind to the 30S subunit. Now remember, the next step is that as you picture the ribosome shifting to the next codon to read it, GGA, it's actually the messenger RNA that moves down the ribosome, but it's easier to show the transfer RNA moving. So anyway, as this shifts to the next codon, this transfer RNA on the A site moves to the P site, and not where it's holding the peptide chain. That opens up the A site for a new transfer RNA to come in and line up to the next codon. But what happens here is the drug basically gets in the way of that translocation, and the transfer RNA on the A site never moves to the P site. It gets knocked off the ribosome. So the transfer RNAs are coming in, and by anticodon sparing the codon, inserting the amino acid, but the transfer RNA never moves from the A site to the P site, so no amino acids are inserted, and that blocks protein synthesis. And another proposed mechanism for some of the aminoglycosides is that when they bind to the 30S subunit, they distort the codon under the A site. If you take that look at that codon CGC in orange there, kind of picture that once the drug unwinds, that codon wraps around it and becomes U-shaped rather than a straight line with the G coming towards you. Well, what happens by distorting the codons is that when the anticodon on the transfer RNA, the GAG, goes to line up, it only has to match up the ones on the two ends. Guanine pairs with cytosine. Now, normally, this transfer RNA would not line up here because adenine doesn't pair with guanine in the middle. But because that guanine's knocked out of alignment, the middle base doesn't have to hydrogen bond. So only the two on the ends have to hydrogen bond. And since many transfer RNAs have two of the three bases in common, what this does is increase the chance that the wrong transfer RNA will insert the wrong amino acid there. Because normally this codon CGC codes for arginine. But because the A and the G in the middle no longer have to match up, only the two outside ones have to match up. GAG actually codes for leucine, carries leucine, and that could insert a leucine by mistake. Now this is actually easier to follow on the animation here, showing misreading of codons. So once the 30S subunit attaches to messenger RNA and the 50S attaches, the aminoglycosides are now attaching to CGC, which is the codon for arginine. Now, normally a transfer RNA with a GCG anticodon would hydrogen bond there. And that transfer RNA with a GCG anticodon would carry arginine. But because this is distorted here, picture U-shaped, the transfer RNA only has to match the two on the end. So here's a transfer RNA that's GAG anticodon. Guanine pairs with cytosine on the ends, but because this G is out of alignment, the A no longer has to match, and so it's the wrong anticodon for that codon, carrying the wrong amino acid, but because all three bases don't have to match now, that increases the chance of inserting the wrong amino acid. So by distorting the codons, where only two of the three bases have to hydrogen bond, that greatly increases the chance that the wrong transfer RNA is going to line up carrying the wrong amino acid. Instead of all three bases on the anticodon matching, this should be GCU. Now a CAU can also attach there. So those are a couple theories as to how the aminoglycosides work. By attaching to the 30S, they're either interfering with the reading of a codon or blocking the ribosome so the transfer RNA can't move from the A to the P site. Now the tetracyclines, which we've mentioned a number of times during the semester, bind reversibly to the 16S RNA in the 30S subunit. And by binding reversibly, they're static rather than cidal in action. But these are thought to actually just block the codon so the transfer RNA anticodon can't get close enough to hydrogen bond. So it's not necessarily just distorting one of the bases in the codon, it's blocking the codon so the anticodon on the transfer RNA can't get close enough to form hydrogen bonds, as we see in this animation. So tetracyclines also bind to the 30S, and it's binding right here on the 30S. But you can kind of envision this fact that now the transfer RNA that has a complementary anticodon, GCG, can't get close enough to form hydrogen bonds because the drug's in the way. And so the transfer RNA never lines up, and all the message can be keep being read. The drug's preventing the anticodons from pairing with the codons, so no amino acids are inserted, and again, no proteins are made. If we look at macrolides like erythromycin, azithromycin, excuse me, these bind reversibly to the 23S ribosomal RNA of the 50S subunit. And again, there's a couple of different proposed ways these work depending on the particular macrolide. One of the things they do is they bind to the enzyme peptidyltransferase. Now, this is one of the proteins in the bacterial 50S subunit of the ribosome, and it's the enzyme that forms the peptide bonds between the amino acids. As the amino acids are being transferred from the P to the A site, transferase, this forms a peptide bond between them. But the drug binds to that enzyme, and so no peptide bonds are formed. And we can see that in this animation. So again, once the ribosome assembles around the messenger RNA, some of the macrolides will bind to the 50S subunit up in here to the peptidyltransferase enzyme. So now the transfer RNAs come into the A site, the anticodon pairs with the codon, but no peptide bonds form between the amino acids. So now the transfer RNAs are reading the message okay, but no peptide bonds are formed between the amino acids. And the other way they work is similar to the way some of the aminoglycosides work. They knock the transfer RNA off of the A site so it never moves to the P site. So they can bind in this case, though, to a 50S subunit rather than a 30S. But again, they're preventing the peptidyl transfer RNA located on the A site from moving to the P site. So here we see a different macrolide. It binds to the 50S subunit, but it binds in such a way that as the ribosome and the messenger is getting ready to read the next codon on the messenger RNA, once that moves into the A site, then remember this transfer RNA, called the peptidyl transfer RNA, moves from the A site to the P site, and the one on the P site exits. But in this case, it prevents the transfer RNA from moving from the A site to the P site, so the transfer RNAs are lining up, carrying the right amino acid, but then they never move from the A site to the P site for translation to continue. The oxazolidinones interfere with translation, and one of the thoughts is that they bind to the 50S ribosomal subunit and interfere its binding with the initiation complex. That is the combination of messenger RNA, transfer RNA, and the transfer RNA matching the start codon AUG. So remember at the beginning, a 30S subunit attaches a messenger RNA, a codon that matches the start codon, AUG, lines up, and we call that the initiation complex. At that point, a 50S subunit can attach. But one of the thoughts is that the oxazolidinones can bind to the 50S subunit and interfere with its attachment to the 30S subunit. So you have an incomplete ribosome form. There's no 50S subunit there to help translate it. So what we're just trying to point out there, you don't have to know all the specific ways, just be able to describe one of those and know in general what it's doing. But the idea there is that how the drug works depends on where it binds on the ribosomes. It interferes with whatever the normal function is at that location where it binds. The streptogrammins, our final group, we have a couple of synthetic drugs that are combined here. They bind to two separate locations on the 23S ribosomal RNA of the 50S subunit. So they're actually blocking two different points of translation simultaneously by combining them. And they do work synergistically, where when they combine them, it works better than if only one or the other was being used. And there's a couple thoughts on this. They might inhibit the attachment of the transfer RNA to the A site, or they may prevent the exit tunnel from the E site from allowing the old transfer RNA to exit. But one way or another, they're blocking two different locations on the ribosome and thus blocking translations. So we have a whole lot of antibiotics that actually work that way. If we go back to our overall chart, these are the ones that bind to ribosomes, blocking translation, causing faulty protein synthesis. Those that bind to the 30S subunit, like the aminoglycosides and the tetracyclines. And those that bind to the 50S ribosomal subunit, like the macrolides, chloramphenicol, oxazolidinol, streptogrammins, and a few others. And there's a little self-check on those that bind to ribosomes. Now, this final section on the modes of actions for disinfectants, antiseptics, and sanitizers, although you do have some definitions under control of microorganisms that you do have to know, notice that this particular lesson has no definitions on disinfectants and the antiseptics and sanitizers, because we are going to cover that in a lab later in the semester. So again, all you have to remember to do on this SoftChalk lesson is answer the performance objectives or the detailed learning objectives for this lesson. But I've included them here because, again, these are chemicals we use to control, but we're going to cover how the disinfectants and antiseptics and sanitizers work in lab rather than lecture at this point. And finally, there's our self-quiz on the whole SoftChalk lesson on how the chemical agents we use affect bacteria. And in our next SoftChalk lesson, we'll look at how bacteria become resistant to those control agents.